Our next speaker is going to be an in-person talk. It's Corday Selden. Corday is a postdoctoral associate here at Rutgers University. She received her PhD in oceanography from Old Dominion University. So her doctoral work was focused on the ocean's nitrogen cycle. Her postdoctoral work here at Rutgers is focused on understanding how protein structure fractionates the stable isotopes of transition metals. So, Corday, take it away.
All right, so as Nathan said, I'm Corday. I'm a postdoc here at Rutgers. And today, I'm going to talk about a set of experiments I've been working on, focused on amino acid binding as a mechanism for fractionating metal stable isotopes. I want to take a few moments to talk about why those among us who fall on the geoscience end of the spectrum should care and think about amino acids binding metals, and the potential power of metal stable isotopes as tools for investigating biological function. Transition metals like copper, iron, and zinc are integral to biological function because they act as protein cofactors. I'm going to give you a few examples of this, but first let me orient you to my plots here. So in red, we see the protein ribbon structure, our polymer of amino acids. And in yellow and blue, we see the sulfur and nitrogen atoms within that polymer, respectively. This is the active site of the protein ferredoxin, which plays a key role in the photosynthetic pathway in mediating electron transfer. And what we can see here, and what I want you to take away, is that iron is at the heart of this reaction. Transition metals can also provide proteins with structure. So this is a classical protein motif called a zinc finger, in which a flexible loop region on the left is affixed to the more rigid alpha helix on the right. And transition metals can also act as carriers for other molecules. So here we're looking at the binding site of the protein hemocyanin, a protein whose function it is to move oxygen through the bloodstream of invertebrates. And we can see that the copper is playing a central role in binding the oxygen molecule. And what I want you to take away here is that within the context of proteins, transition metals are bound by amino acids. And there's a core set of amino acids that play a particularly important role. These include cysteine, which has a thiol residue, and histidine, which has an imidazole ring. These sites are also very structurally diverse, and this is a theme I'll return to later on. Many transition metals have multiple stable isotopes, which are fractionated by biology. Today, I'm primarily going to talk about copper, which has two stable isotopes of mass 63 and 65. And for those of you who are on the other side of the spectrum and maybe don't think as much about stable isotopes, I'll remind you that when we talk about stable isotope abundances, we do so using delta notation. And this is simply the ratio of the heavy isotope to the light isotope relative to a standard. So when I show you delta values that are less than 0, this means our sample is enriched in the light isotope relative to a standard, and the reverse is true for those values above 0. I'll also show some cap delta values, and this is very simply the difference in delta value between two measurable states. So in the context of my experiments in a moment, this will be the difference in delta value between our complexed copper and our free copper. Biology fractionates copper isotopes relative to the growth environment. Here, we're looking at those cap delta values for copper for organisms relative to their environment. So in the case of the animals, the aquatic animals, which are the first arrow, this is relative to the mean copper, the mean delta value of ocean seawater. And what I want you to take away here is that we see a wide range of fractionation effects associated with uptake, both in bacteria and in single-celled eukaryotes like yeast, and also in animals. Now, many of you may be more accustomed to thinking about carbon or nitrogen or oxygen isotopes, and these values may seem small, right? Metals are lower down on the periodic table, they're heavier, so the relative difference in mass between their isotopes is smaller, and we see smaller fractionation effects. But let me assure you that with current analytical techniques, we're able to distinguish these values within about 0.05 per mil. Copper isotope abundances can also vary among tissues within the same organism. I've switched up the x-axis on you here, so now we're simply looking at delta values, not the difference. And what we can see is that in healthy humans, serum, the liquid portion of the blood, so without the clotting factors in the cells, is isotopically lighter than brain tissue. So we have fractionation happening within an organism. And these values are altered with disease state. So in patients of ovarian cancer and liver disease, serum copper isotope values become isotopically lighter. And this is true too of human cortical tissue in Alzheimer's patients. And I will point out that while the range that we see among the populations that were sampled in this study are broad, these are statistically significant differences, meaning that they can potentially be used as a diagnostic indicator. But more importantly, for our purposes and interests here, it suggests that copper isotope fractionation depends on the metabolic function of the organism. Ultimately, the molecular mechanisms which underpin this variability remain very poorly understood, and this really complicates our ability to interpret biological isotope effects and to discriminate among biological and abiotic isotope effects. However, understanding the principles governing this fractionation is essential to leveraging these types of observations. Myriad processes at the subcellular level have been proposed to regulate metal isotope composition. These include surface processes like adsorption and reduction, uptake, and intracellular partitioning, by which I mean the distribution of metals among proteins with distinct binding environments. And while the first two of these boxes have garnered much attention, particularly in the geologic literature, we really need to invoke this third box in order to explain the sum set of observations which we have about not just copper isotopes, but other metal isotopes as well. Now, I want to give you an example of what I mean by that. Here, we're looking at delta values for two proteins isolated from human cortical tissue, from brains. On the left-hand side, we have metallothionein. This is a copper storage protein. It's often expressed in order to deal with copper toxicity under high copper concentrations. And superoxide dismutase, which is involved in oxygen radical detoxification, the dismutation of superoxide. And what Larner and colleagues found in this study was that metallothionein is isotopically light relative to superoxide dismutase. And this type of natural variability among proteins has been observed elsewhere. So here we're looking at delta values for those same two proteins, but this time in Saccharomyces, yeast. And what I want you to note is that the difference in the delta values between these two proteins is relatively consistent, suggesting a consistent underlying process. Ab initio calculations based on density functional theory predict that amino acids at metal binding sites differentially fractionate copper isotopes. And this suggests that differences in copper isotope composition among proteins may relate to the local coordination environment. And I'll point out here that metallothionein binds copper using the sulfurs of cysteine, while superoxide dismutase binds copper using those nitrogens in blue in the imidazole rings of histidine. While this is a very compelling hypothesis, there's currently no experimental data to test these predictions, which is where this study comes into play. The most common amino acids which coordinate copper in metalloproteins are cysteine and histidine, which we've already discussed, and glutamic acid, which has a carboxyl functional group. Ab initio calculations suggest that cysteine will preferentially bind the light isotope of copper, while glutamic acid and histidine will have relatively little isotope effect. Our objective in the study, which I'm going to present here, was to investigate copper isotope fractionation due to amino acid ligation. So does the identity of the amino acids at our copper binding site result in isotopic fractionation? We tested the isotopic fractionation of copper by amino acids using an equilibrium Donin dialysis system. So the system consists simply of two Teflon cells separated by a cation permeable membrane. And the thing to remember about this system is that the membrane permits the passage of free copper, but not of bound copper. So the bound copper, our copper complexes, will be retained in whichever chamber we place them in. To test the equilibration time of this system, I made up solutions that contained free copper and another that contained only the background constituents. These were placed in the separate chambers, and the system was allowed to equilibrate through time, right? So again, the free copper is able to come to equilibrium as it can permeate through the membrane. And we see this reflected in this plot. So on the x-axis, we have dialysis time, the amount of time that our solution spent in the system. On the y-axis, we have the copper concentration of the solutions in the donor in that blue chamber and the acceptor in red. We can see that the system equilibrates within 24 hours. This was true not only of mass equilibrium, but also of isotopic equilibrium, with the delta values shown here as red and blue X's for the two chambers. And you'll note that they're within error of one another. We tested the isotopic fractionation of copper by different amino acids using the same setup. And here we're simply adding our amino acids to the donor solution. Again, that free copper equilibrates, our bound copper is retained in the donor, and that's what we see reflected in this plot. On the x-axis, we have our 4 experiments, our control, and our trials with cysteine, glutamic acid, and histidine. On the y-axis, we have the mean copper distribution in the 2 chambers, and this is the mean of three replicate chambers. And the thing to note here is that in the trials with cysteine, glutamic acid, and histidine, more copper was retained in the donor solution than in the control. So this suggests that there was ligation occurring, that we formed those copper amino acid complexes. This was more so the case for cysteine, which is a very strong binding agent. We measured the delta values in all of these solutions, and I calculated the del 65 copper value of our complex copper using a mass balance equation. Here on the x-axis, we have cap delta values. So again, the difference between our complex copper and our free copper. On the y-axis, we have our 3 amino acids. The red boxes indicate those ab initio values. The black dots indicate the results of individual dialysis experiments with propagated analytical error shown as error bars. And what we can see here is that glutamic acid imparted very little isotopic fractionation as expected, but that cysteine and histidine had larger magnitude isotope effects than predicted, although the directionality of these effects was consistent with theory. So what we can take away from this is that amino acid binding does indeed fractionate copper isotopes. And this can help explain some of those observations, including the ones I showed at the beginning. You'll remember that metallothioneine with its sulfur-heavy binding site was isotopically light. Superoxide dismutase with its nitrogen-rich binding site was isotopically heavy. This work suggests that the relative abundance of proteins with nitrogen and sulfur-rich binding sites within a cell can alter the cell's overall copper isotope composition. If this holds true for other metals, it strongly suggests that the metalloproteome, so the sum set of metal binding proteins in a cell or a population of cells, can affect metal isotope distribution at higher levels of biological complexity. By extension, if fractionation of metal isotopes by cells can affect the distribution of metal isotopes in populations of cells, then it may also manifest in biogenic material and in the environment. And with respect to the astrobiological implications of this work, which I know is why we're all here today, I'll remind you that metals persist in the environment long after other lines of biological evidence have deteriorated. Provided that these metal isotope signatures can be preserved, so provided that they are not overprinted by other isotopic fractionation processes and can be disentangled from the record, they represent a potentially powerful tool for investigating metabolic function through time and space. But before we can begin to apply this tool to astrobiological questions, we first need to better understand the extent to which the metalloproteome controls the isotopic composition of individual cells and of cells in the modern environment. And with that, I would like to just take a moment to thank my collaborators, Catherine Schilling and Nathan Yee, who were instrumental in the design and execution of this work, Linda Godfrey, who helped me with the copper isotope measurements, and Anirban Basu Naomi Saunders, with whom I'm working to conduct similar experiments with zinc and nickel. And of course, to Paul and Vic and Bea and the entire ENIGMA team for your support. And with that, I'll take any questions.
Let's thank Corday one more time.